Ladies and gentlemen, welcome to the announcement of the Brain Prize winners 2020. Also, a warm welcome to Lena Skola, who will now give a short introduction to the Brain Prize and reveal the winners.
Ladies and gentlemen, viewers from around the world, I'm Lena Skola, CEO of the Lundbeck Foundation. The past couple of years, I've had the privilege of welcoming you to the announcement of the Brain Prize winners. It's always been an exciting day, and this year is no exception. But the circumstances have changed. The COVID-19 pandemic has cost thousands of lives, put some of the most advanced healthcare systems under pressure, and shut down entire societies. Researchers around the world are working hard to develop a vaccine, while healthcare professionals are struggling to save lives. Nothing is business as usual, nor is this announcement. It takes place only digitally. There is no audience, and there are no prize winners here in person. But that does not mean that we cannot, with the proper distance and with certain precautions, honor a new group of outstanding neuroscientists with the Brain Prize 2020. After all, it is the 10th year anniversary of the prize. For 10 years now, we have been awarding the world's greatest brain research prize to some of the brightest minds, acknowledging their transformational scientific breakthroughs and paying tribute to their talent, their hard work, their dedication, and their brilliant ideas. The Brain Prize has come a long way in those 10 years, evolving from a new and unknown award to a globally recognized and highly esteemed prize, progressing from purely European to international, and starting with none, and this year reaching a total of 34 winners. But why did we decide to establish this prize more than 10 years ago? Some would argue that the world has enough scientific awards already. Well, the brain is a key focus area for us, and we believe there is nothing more important nor fascinating than the brain. When the brain is not functioning properly, the cost to the affected patient and their relatives is huge. It's our most vital and complex organ, not merely responsible for a number of bodily functions, such as speech, thoughts, and communication, but it's also the home of the human identity. Therefore, understanding the basic mechanisms of the brain will not only lead to better insight into the human mind, understanding why we are who we are, but also to better treatment of brain diseases, which is ever so important. That is why honoring and supporting the work of researchers who make groundbreaking discoveries about the brain is so important to us. And that is why we established the prize, to honor those who devote their brains to helping ours. And now, it's time for what we've all been waiting for, the announcement of the 2020 Brain Prize winners.
Between the many qualified nominations from all over the world, the selection committee chose to award the Brain Prize 2020 to Professor Huda Zogby, Baylor College of Medicine, USA, and Professor Sir Adrian Bird, University of Edinburgh, UK. And now for the motivation.
Professor Huda Zogby and Sir Adrian Bird have been awarded the Brain Prize for 2020, worth 10 million Danish kroner for their pioneering work on Rett syndrome. Their work has established the importance of epigenetic regulation in both brain development and the maintenance of normal adult brain function. It also points to novel opportunities for the treatment of this and other neurodevelopmental disorders.
To enlighten us even more today, we have science journalist Line Friis Frederiksen with us, who is going to interview the chairman of the Brain Prize Selection Committee, Brain Prize laureate, and Professor Richard Morris.
Hello everyone and welcome to the announcement of the Brain Prize 2020. And also a big welcome to you, Professor Morris. Nice to see you.
Hello, Lena, and nice to see you too. Where do we start?
Well, I was wondering whether you could please start by giving us just a short overview of why the selection committee chose exactly these two winners for this year's Brain Prize.
Well, as you've just heard, the Brain Prize for 2020 is awarded for fundamental pioneering work on a disorder affecting children called Rett syndrome. Adrian Bird and Huda Zogby identified alterations in a specific protein called MECP2 as the cause of this brain disorder. And it was shown in their research to recognize methylated DNA and to participate in controlling gene expression. Their novel findings, including successful genetic treatment of the condition in animal studies, have established the importance of epigenetic regulation in both brain development and the maintenance of normal adult brain function. And so these observations raise the fascinating question of whether neurological disorders of early childhood are always reversible, perhaps not, and thereby point to opportunities for ameliorating the symptoms of not only Rett syndrome, but hopefully other neurodevelopmental disorders, including autism, and do so using gene therapy.
These are indeed amazing perspectives. But before we go into more details with that, could you maybe shed a little light on how the selection committee actually find the winners? I mean, it's probably not an easy task.
No, not easy at all. In fact, each year the selection committee for the Brain Prize is faced with deciding amongst over 100 nominations, all of whom have done amazing biomedical science, as to which one or which set of them is most deserving of the prize that year. So we read the nominations, the scientific papers that the nominees have published, and we take up references on what we judge to be the best candidacies. And then we meet on two separate occasions and discuss and decide. It's as fascinating an intellectual exercise to carry out as one might imagine. It's also actually difficult to decide on the winners.
And now, Professor Morris, could you tell us more about the disease, the Rett syndrome, that Professor Adrian Bird and Professor Huda Zogby has been the key to gain an understanding of?
Well, Rett syndrome was first described in Vienna in 1966. And it's a devastating genetic disorder that typically becomes apparent after 6 to 18 months of age in baby girls. And the symptoms include problems with coordination and repetitive movements, later with language development, often the slower growth, problems with walking and a smaller head size. And additional complications can include seizures and sleeping problems. Its prevalence is about 1 in 8,000 females, which means there are likely as many as 700 cases here in Denmark. Because the MECP2 gene is on the X chromosome, girls carrying a Rett mutation have brains that are mosaic for the mutation, meaning that some cells express the mutant protein, while others express normal protein. By contrast, in males, all brain cells are affected, and so the condition is lethal in males before birth.
So indeed a very severe disease. And could you now, Professor Morris, tell us more about the two Brain Prize laureates? Who are they? Tell us about their scientific journey.
Well, our two laureates this year are Professor Sir Adrian Bird, the Buchanan Professor of Genetics at the University of Edinburgh in the United Kingdom, and Professor Huda Zogby, an investigator of the Howard Hughes Medical Institute and also Professor of Pediatrics and Human Molecular Genetics and of Neurology and Neuroscience at Baylor College of Medicine in Texas in the United States. Professor Bird's English by birth, but has worked in Scotland for many years. Professor Zogby is Lebanese by birth, but now a naturalized citizen of the United States, where she's lived and worked since graduating from university in her native country. And these two are respectively an outstanding molecular biologist with interests in the nervous system and a remarkable medical scientist whose research has focused on a major issue, developmental disorders of the nervous system. Now, the first step of their story was pioneering research by Adrian Bird, who in 1992 was the first to discover this protein called MECP2, which reads a DNA methylation signal. And at the time, Dr. Bird had no knowledge of the clinical importance of this protein. He was simply pursuing his discovery research program focused on understanding facets of gene regulation. Now, Dr. Zogby, who received her medical training in the United States and began her career as a doctor in Texas, serving as a pediatric resident, and the opportunity to do research in pediatric neurology as well led to her discovering the gene responsible for spinocerebellar ataxia, another condition. Her attention was then drawn to trying to find out what had gone wrong with a cohort of girls presenting with a very severe neurological disorder. And her determination and resolve led her to discover in 1999 that the affected girls all had mutations in the gene encoding Adrian Bird's MECP2 protein. So propelled by the discovery of MECP2 mutations as the cause of Rett syndrome, parallel streams of work in both MECP2 and this devastating neurological disorder went back and forth between these two main research groups, leading to several striking advances. Adrian Bird and his colleagues showed, for example, that MECP2 is exceptionally abundant in mature brain cells. And it's also defined by two key interactions, with methylated DNA on the one hand, and on the other with a gene silencing complex. And this led Dr. Bird to propose that MECP2 normally delivers the gene silencing machinery to methylated sites in the genome. And in turn, Dr. Zogby demonstrated that the ability of MECP2 to form a bridge between these macromolecular partners is disrupted in clinically significant Rett mutations. So Huda Zogby's discovery of the link between MECP2 and the disease led both research groups to carry on their pursuit of the fundamental molecular and neural basis of the disorder. And each group then created animal models of Rett syndrome. Dr. Bird knocked out the MECP2 gene, and he found that the mice he'd created closely mimicked many of the key features of the human disorder, including tremor, immobility, breathing arrhythmia, and late-onset phenotypic features in females. Dr. Zogby found that the amount of MECP2 in various patients was related to the severity of their symptoms. In fact, surprisingly, not only is not enough MECP2 problematic, so also is having too much. So she then generated mouse mutants bearing the mutations found in the human patients, with studies in both her and Adrian Bird's group establishing that MECP2 deficiency in the brain is the sole culprit. Its absence or its altered expression is responsible for all the obvious phenotypic effects of the disease.
So, as almost always in science, it's really not a straightforward way, and you have no idea of the goal when you set out on your scientific journey. But the question that of course comes to mind is, with this new detailed insight into the Rett syndrome, is there a hope for a treatment or a cure for the affected individuals and their families?
Well, that is indeed a very important question. What about correcting the genetic abnormality? Well, it turns out that Rett syndrome doesn't cause brain cells to die in humans, nor in mice. So with the cells still alive, Bird's group therefore asked whether the phenotype could be reversed and brought back to normal by providing again the missing protein long after onset of the phenotype. And astonishingly, activation of a quiescent MECP2 gene corrected all the measured phenotypes: male survival, motor function, breathing abnormalities, and altered neuronal morphology. And in parallel, Huda-Zoghbi showed that the disease can be triggered when, after a period of completely normal development, MECP deletion is induced in adult mice. And so she was also able to dissect some of the neural mechanisms underlying the syndrome by showing that MECP2 mutations in different brain regions are responsible for distinct aspects of the disease. So this astonishing set of gene rescue studies have helped promote a fundamental change in attitudes to neurodevelopmental disorders in humans that have long been assumed to be tragically irrevocable. Because, the crucial thing is that these new findings suggest that Rett syndrome may be treatable. With the mice having confirmed the therapeutic potential of gene therapy, clinical trials in humans are actually imminent. And the results that will come from these trials are of course, eagerly awaited, not least by the parents of affected children whose lives have been turned upside down by discovering, after about a year of normal development, that their daughter has the condition.
Let's really hope for a cure. And now, are there any closing remarks you, as the chairman of the selection committee, would like to add to the amazing story of the Brain Prize 2020?
Well, I think maybe just that the Lundbeck Foundation looks upon this still evolving scientific story as a truly remarkable one. An elegant and triumphant combination of basic science and clever clinical observation brought together by modern genetics as applied to the nervous system. So I and my fellow members of the committee offer our congratulations to both Professor Huda-Zoghbi and Professor Adrian Bird.
That is all for this year's Brain Prize announcement. We hope to see you all next year, and until then, stay safe.